So, as a graduate student here in the Bates Lab in the Department of Microbiology, we study how viruses interact with their hosts, especially at the cellular level. We're really interested in understanding what the specific cellular factors are that are required by the virus, with the hope of then understanding the virus biology and then also identifying novel targets for antiviral therapy. And so in order to do this, we utilize an approach that really lets the virus tell us what it needs. So using a population of cells that we've mutagenized so that they contain about 1 to 3 mutations per cell, we then infect those cells with virus, and this ends up killing about most of the cells. However, the cells that do survive do so because of mutations in genes that are essential for that virus. And so we can identify those mutated genes using DNA sequencing and then use those genes identified to guide our future studies. And so we've done this with a number of viruses, including many bunyaviruses, our RNA viruses, including Andes virus as well as pumila-virus. And while I was a graduate student, I was really interested in this newly identified virus, the severe fever with thrombocytopenia syndrome virus, which was identified to cause disease in people in China, South Korea, as well as Japan in the last decade. And so from our screen with SFTSV, we identified this gene that's involved in the formation of glycolipids, which are lipid molecules that have sugars appended on them. And so if we block the synthesis of these glycolipid molecules, we ended up blocking infection with that virus. And we found that it was affecting a very early step of the virus at the level of virus entry. And so if we followed the virus into the cell using a high-resolution microscopy, we could see that the virus was accumulating within these structures in the cytoplasm. And so this was likely due to the virus's inability to escape from membranous complexes within the cell, and therefore unable to release its genetic material into the cytoplasm in order to replicate. And so from these studies, we are excited to learn something new about this pathogenic virus and its biology within the cell, as well as identify a new target that can potentially be used for antiviral therapeutics. And so some of the compounds that we actually used in these studies are already FDA approved for treatment in humans of people who have disorders related to these glycolipids. And so with that, I'm super honored to have been chosen as the Fraser Graduate Student Awardee this year. Thank you so much to the Cell and Molecular Biology Graduate Group, as well as the Department of Microbiology, and of course Dr. Paul Bates for all of your tremendous guidance and support during my time in graduate school. Thank you.